Hello, and welcome to the Scientist.com webinar series, The 3 Rs of Animals in Research. This is part 2, Reduction, and it's presented in collaboration with the NC3Rs and Hera BioLabs. My name is Megan Loy. I am the Category Director of In Vivo Services here at Scientist.com, and I've been with the company since December of last year. I have a master's in biotechnology from the University of Wisconsin-Madison, and I've been working at Scientist.com to help support the in vivo categories, including toxicology, DMPK, and pharmacology, and advance animal welfare compliance. Scientist.com is a B2B marketplace, and we were founded in 2007. We currently have headquarters in San Diego, but we have offices around the world, including Boston, Cambridge, UK, and Tokyo. We have about 70 full-time employees currently and over 3,000 global suppliers, covering 4,000 or more research services in our network. Scientist.com is an online marketplace for R&D outsourcing. So that means we connect research organizations of any size with a global list of suppliers of custom R&D services. And you can see some of these services down at the bottom, but there are many more available. One of the things we do here is support research, and that means that we support animal welfare as well. So animal research is essential in pharmaceutical development, but it's our obligation to conduct research thoughtfully. And so we support efforts to replace animals, reduce their numbers, and refine procedures wherever possible. We also support animal welfare efforts through our supplier due diligence program, through animal welfare information gathering and compilation, and through educational programs such as this one. Our 3Rs webinar series is continuing in 2 weeks with refinement featuring Sinclair Research. If you'd like to hear any of the previous webinars, please go ahead and register or request the recording from us. And now it's my pleasure to introduce Dr. Sam Jackson. If you have any questions for Sam or any of the other speakers, please go ahead and post them in the Q&A session at the bottom of the screen. Sam is the program manager at the NC3Rs, and he examines the potential to replace, refine, or reduce animals used to model disease, to measure efficacy, and to examine the safety of new drugs. So this includes application of new technologies and human tissue and cells to replace animal models, improve productivity to humans, and to benefit disease research and drug development. He's recently organized several workshops and surveys that explore how human tissue is used in cancer research and safety pharmacology, and identify the barriers to increase use of the resource. So prior to joining the NC3Rs, Dr. Jackson worked for 10 years in research, doing postdoc posts in neurobiology in both academic and industry settings. And without further ado, I'd like to introduce Dr. Sam Jackson.
Great, thank you very much, Megan. So I am just going to introduce the topic of for today. I'm going to tell you a very small amount about where I work at the NC3Rs, a little bit about the 3Rs in general, and then give you some examples of reduction. So just a little word about where I work. The National Centre for the Replacement, Refinement and Reduction of Animals in Research is based in the UK. And following on from a government consultation in 2002, recommendations were to give greater priority to the development of non-animal methods and the 3Rs generally, and to set up a national UK centre for the 3Rs. So in 2004, we were established really to lead the UK's 3Rs agenda. And since then, we've become a world-leading authority in the area. We really apply the 3Rs as a framework to support science and innovation and improve animal welfare through investing in people and practice through our grant funding programs, through driving commercialization of 3Rs relevant technologies through our open innovation scheme, Crack It. And to do so, we work across the bioscience sector with academics and industry companies involved in biomedical research who are using animals and alternative methods. We also work with a wide range of other stakeholder organizations outside of biosciences, including in the chemicals, agrochemicals, and personal care product sectors. We receive around £10 million a year, which we spend on our research funding schemes to support open innovation platform and to carry out in-house activities around specific subject areas. And we often use working group or data sharing approaches to achieve these aims. We are around 30 staff based in the London central office, and we have 5 regional posts around the UK as indicated on the map here. And we also work very closely with scientists in Europe and in the US as well. So just as a background to the 3Rs, as I mentioned, we at the NC3Rs think of the 3Rs as a framework within which to do biomedical sciences. Today we're going to be talking about reduction. But the 3Rs really are important for multiple reasons. So first off, they provide an ethical framework within which we can critically assess animal use for use in science. 3Rs are enshrined in legislation. So the Animal Scientific Procedures Act in the UK and similar EU and US legislation defines the 3Rs as one of the ways that we should be thinking about using animals in research. There is great public support for the 3Rs. So in the UK, an annual poll, Public Attitudes to Animal Research, indicates that the public support animal use in research only when the principles of the 3Rs are adhered to. And finally, the 3Rs can be implemented to improve scientific practice and to build new businesses. And this is part of our role in the UK. So today we'll be talking specifically about reduction. Reduction is defined as methods which minimize the number of animals used per experiment. However, the NC3Rs, we think about this as being appropriately designed and analyzed animal experiments that are robust and reproducible and truly add to the knowledge base. So I wanted to give you some examples of reduction. We're going to hear an excellent case study later on, but just to give you an idea of the breadth of what can be considered reduction type technologies or approaches. So reduction refers to methods that minimize the number of animals used per experiment or study, consistent with scientific aims. It's essential for reduction that studies with animals are appropriately designed and analyzed to ensure robust and reproducible findings. And the examples I have on the slide here are using improved statistics and experimental design, and I'll give you an example of our experimental design assistant in a second. Reduction also includes methods which allow the information gathered per animal in an experiment to be maximized. This is in order to reduce the use of additional animals. Examples of this include using some imaging modalities which allow longitudinal measurements in the same animal to be taken, for example, rather than culling cohorts of animals at specific time points. Things such as microsampling of blood, where small volumes enable repeat sampling in the same animal. And reduction in these cases has to be balanced against any potential additional suffering that might be caused by their repeated use. And finally, falling into the reduction category are ways of sharing data and resources, so either animals, tissues, or equipment between research groups and organizations, and this can contribute to reduction. So some of the benefits of applying reduction to research include improving the accuracy and validity of the data collected from the animals. So a well-designed and executed study can ensure that animals are used in the most effective and efficient way possible. This is more likely to result in a valid data set containing useful translational information we can then take through to inform the clinic. It can also increase the fidelity and granularity of the data collected from a single animal. So collecting more data from one animal can increase the power of a model and potentially reduce number of animals that way. So for example, applying new technologies to longitudinal studies or increasing the sensitivity and specificity of an assay or a measurement technique. And these really do have the potential to save time and money, as well as reducing animal numbers that are used in experiments. So I'd like to just give you two quick examples of resources or tools really that we have come up with at the NC3Rs, which can help with this area. So first off, I'd like to highlight the Experimental Design Assistant, or the EDA. And if you'd like to see this for yourself, the URL is at the top of the page here. So the EDA was developed to improve the design of animal experiments. It's a web application with an extensive supporting website, which helps researchers design their animal experiments better. The target audience really are early career researchers, so PhD students and earlier postdocs. It enables users to get a greater understanding of experimental design and especially how it might impact their particular experiment. And you build up a diagram of the experiment on the screen, and the system will feed back to you to improve the experimental design and suggest things like randomization, blinding, and help you with sample size calculations. You can share these diagrams around as well, so this can improve transparency and help communication of experimental design. So the EDA was developed by collaboration from a working group of in vivo researchers and statisticians, and it's been designed by a software company specializing in artificial intelligence. The second resource I wanted to highlight were the ARRIVE research reporting guidelines. So these were developed as part of an NC3R's initiative to improve the quality of reporting of biosciences research using animals. It's essentially a checklist of 20 items, which are the key information which are necessary to describe a study comprehensively and transparently so that it can be repeated by somebody else. It can be used to ensure the reproducibility of animal research and avoid unnecessary animal use. And the guidelines have been endorsed by a wide range of journals, some of which require a completed checklist on acceptance of a manuscript. So this is becoming more embedded in the publishing process. So that's all I have to say for my introduction. I'll pass back to Megan. Thank you.
Thank you, Sam. And for the attendees, I'd like to remind you that if you have any questions for Sam, please go ahead and submit them in the Q&A portion at the bottom of the screen. And now I'd like to introduce Dr. Seton Jamling from Hera BioLabs. Dr. Jamling is a molecular biologist, and he specializes in custom rodent models. He's the VP of Research and Development at Hera BioLabs, and he's developed the SRG OncoRat, which is a double knockout immunodeficient rat model for xenograft studies, and he'll be discussing some of this today. Without further ado, I'd like to hand it over to Dr. Seton Jamling.
Thank you, Megan, and thank you, Dr. Jackson, for the overview. My name is Seton. I'm the VP of Research and Development here at Hera BioLabs, and we are a young, small CRO, started in 2015. So in the past 4 years, we have developed a rat model for preclinical oncology studies. And today I would like to share with you how use of that model, can be applied to the 3 R's principle. So without going in detail here, as Dr. Jackson already covered it, 3 R's basically the replacement, reduction, and refinement of animal use in research, which we all deal with. So it's a legally required principle, it's ethically important, and it's scientifically important. And today here, focusing on the reduction part of it, I will give you examples how we use the SRG OncoRat that allow us to minimize the number of animals used for experiment, and also how we use it to gather more information per animal in an experiment that leads to overall reduction of animals. So the SRG OncoRat, as I said, is a RAG2 IL-2 double knockout rat. It has an 8 base pair deletion in the RAG2 exon, which makes it BJJ recombination deficient, leading to absence of B and T cells. And then it also has a 16 base pair deletion in the IL-2 receptor gamma gene, which leads to ablation of the NK cells. And on the right side of the screen here are basically the fact plots for showing that these animals don't have any mature T cells, B cells, or NK cells circulating in the system, and it's hard to find them even in their source organs, thymus and spleen. So I will talk about 2 different utilities of the SRG OncoRat. The first one here being for xenografts, and the example I'm showing here focuses especially on xenografts for cell lines that are difficult to work with existing mouse models, right? Cell lines like VCAP, which is a AR-dependent prostate cancer line, which has poor engraftment efficiency in the NSG mouse or the not SCID mouse or other mouse models. And not only poor engraftment efficiency, but highly variable growth kinetics in the mouse models, leading to very poor enrollment rate into the studies. So either enrollment rate is really low or the enrollment window is too wide, compromising the data, fidelity of the data that you generate. So in the SRG OncoRat, we have consistently getting over 85% engraftment rate, and compared to a variable 20 to 60% engraftment rate in the literature. In our own hands, we can get about 40%, but the growth kinetics in the NSG mouse are so variable that our enrollment window is over a month, a 30-day period. So in the SRG OncoRed, we're getting 85% engraftment rate, and over 90% of them can be enrolled within a 7 to 10-day window for studies. So on the right is showing the discrepancy in the engraftment rate between the OncoRat and the mouse models, and further along the growth curve is being shown, the studies we have done head-to-head, tumors growing in the VCAP cells growing in SRG versus in SG mice. And so these engraftment OncoRats can be utilized for efficacy studies. As an example, on the bottom, it's just a standard of care treatment to enzalutamide in the green line compared to the vehicle control on the red line. As you can see, those tumor growth is really tight. 6 animals per arm is enough to generate really good data in this animal. So in terms of the focus on reduction of the animals used for a study like this, for a tough cell line that engrafts poorly in the mouse. So in the SRG OncoRat, let's say, I'll put some numbers up here, just representative numbers. If you wanted to enroll 32 animals in a study, let's say 8 animals per arm, 4 arms per study. So for the SRG OncoRat, we would want about 34 to 38 animals that engraft and grow the tumor, because we have a really high enrollment rate, which means 85% engraftment rate, we inoculate about 42 animals. Whereas in the NSG mice, if you want to enroll the same number of animals, 32 animals, within a 14-day enrollment window, you would have to get about 32 to 96 animals engrafted, just because the growth kinetics is so variable. And then with the low engraftment rate, we would have to inoculate about 160 animals. This is based on the internal work that we have done, the growth kinetics shown on the right. There are some literature out there where people are getting a little bit better, and that number might be a little bit lower, but there are others that show, that those numbers would be higher also anecdotally to the clients that we are talking about. So that is a big reduction in the number of animals you start with here for an efficacy study. Beyond that, for xenograft studies like that, right, not just running an efficacy study, but the second point of reduction where you try to generate as much data as possible from a single animal. So with the rat being a much bigger organism than the mouse, it has about 10 times the volume of blood that a mouse has. We can do a lot more from a single mouse. So we can do serial blood draws, for example, for PK analysis or for biomarker analysis, as we are showing here. So let's say theoretically we do, we want to look at the biomarkers here. So this is for VCAP study. We're looking for serum PSA level, which is a biomarker for the cell line. And so you say, 8 animals, 2 animals from each arm, and looking at 3 size references as the tumor grows. So for the SRG OncoRat, these can be the animals that are enrolled in the study. We don't have to enroll any more studies, because you can just pick the animals from the group and do serial blood draws as it goes. So the total number of animals remain at 42. Whereas for NSG mice now, this would be a terminal blood draw. For this assay and for many other, ClinPath assays that you might be doing. So you have to inoculate additional animals when you start off. So just keeping the numbers as small as possible. Let's say 8 animals, 3 references, that's 24 extra animals to what we started with, bringing the number up to 184 animals now, where the OncoRat remains at 42. And on the right, as you can see, we did that exactly, blood draws from these animals at different tumor size volumes, serially drawn, and then check correlate the PSA level to the tumor size. And you can see it's a very nice correlation of R value of over 0.9. The tight correlation indicating, the tumor is growing, and it's exactly what it's supposed to be. So that's one. Another thing we have done with the rat is we have done tumor biopsies or sample fine needle aspirates that you could do for pharmacodynamic studies or molecular analysis, target engagement type analyses that you can do. So the tumors in the rat, again, for this, as you saw in the previous slide where I was showing the NSG versus SRG growth curve, the tumors grow so much bigger in these rats. These rats, being physically bigger, are able to support a much bigger tumor volume than the mouse, which allows us to do fine needle aspirates, and the tumor's growth curves remain unaffected with doing fine needle aspirates. So again, let's say 8 animals, 3 time points, we want to look at it. SRG oncoret, again, the number stays put at 42. You can recruit the animals in the study to do these fine needle aspirates without having additional animals. And for the NSG animals, again, you would have to add in at least 24 animals. So, that number goes up, which it could be more because engraftment and growth rate difficulties, but at least you'd need to add 24 animals. So that's on the right. I'm showing you a couple of things we have done with these tumor biopsies. Again, on the Western blot shown here is again for the VCAP cells. We are looking tumor biopsies, and we are doing Western blot to look at androgen receptor expression in these tumors at day 1, day 21, day 28 of the dosing study. And also we're looking for PSA expression in these tumors. As you can see, everything looks good. These tumors are what we think they are. They're expressing the receptor target of choice that we're looking at. And the biomarkers are also there. So, and these animals are enrolled in the study we're looking for. Another type of thing on the right here is for a non-small cell lung cancer we were looking at treated with crizotinib, which is a MEK inhibitor, right? So with a fine needle aspirate, we're able to look at the AKT to phosphorylated AKT ratio in the vehicle control versus the treated animals. So as the tumor size is decreasing, you can also see that the biomarker are getting phosphorylated or not over time, and these can be done at multiple time points. So again, total of these for these 3 stuff. As you can see, the animal numbers we use are down to about a quarter required in the rat as compared to the mouse. So we're happy that we're able to show this utility of the oncorat. And so this is one example. We have done similar studies for other lines also. We'd be happy to share, if you have more questions. So it's a real good tool for xenograft lines that are tough to work with in the mouse, but even if they work well, you can do these combinations, get much data, more data from a single animal, which might, be beneficial for your studies. And not just for animal numbers, but there's also something to be said about the quality and the power of the data that you generate, which Dr. Jackson touched on a little bit, right? So for these kind of biomarker analysis, PK analysis, molecular characterizations, these then, when you use the oncorat, you're generating these data serially from the same animal being monitored over time. And it's the same animal that you also measuring the response to for your drug. So the data, I think, is much higher quality, whereas for an NSG mice now, for each individual time point, as you are already aware, you'll be using a separate animal, and which is, different from the animal that's being treated with a drug. And so over time, these many different animals' data points are being strung together to put on a single line. And so your data quality gets much better when you're using the oncorat. So the next, the second point I want to touch in on for the utility of oncorat in this focus is for establishment of PDX and PDX expansion. So as I mentioned before, and as I show you on the graph down here, the tumor volume, the SRG rats, oncorats can support about 10 times the tumor volume than an NSG mouse or any other mouse could. So as you can see here, these tumors can grow up to 30,000 millimeter cube, whereas in the mouse, they're at about 2,000 to 3,000 millimeter cube in volume. That's your humane endpoint. So which means in the PDX focus, when we grow a tumor, a patient's tumor on the oncorat, from a single animal that grows it, we can generate over 100 2x2 millimeter tumor pieces that can, that go into individual animals. So whereas to generate that in the mouse, you would have to go through several passages to generate that much tumor. So that being the main point here. And then also, as we show for VCAP, this data here on the right for the engraftment efficiency is for non-small cell lung cancer patient samples. So we, the SRG OncoRat again, is displaying much better engraftment rate for patient samples, the non-small cell lung cancer patient samples. And we are trying some other ones, ovarian gynecological ones, the ones that are tougher to grow in the mouse. And so in the mouse, you get an engraftment rate of about 20 to 40% range. If you scan the literature with the SRG OncoRat, we are getting over 80% engraftment efficiency. So combination of, when you get the tissue from a patient, you get a limited number of tissue. You can maybe put them in 2 or 3 animals. So a combination of the engraftment improvement versus how big these tumors can grow to establish a bank of, let's say, about 1,000 tumor pieces. We are estimating about a 90% reduction in the animals used to establish and expand that PDX when you use OncoRat. And I'll show you some data on the right, histology data, original patient sample, the first passage grown in OncoRat, and the second passage, just showing histologically, that tumor is what it's supposed to be and shows the markers that we are looking for. So, right, so we're reducing the number of animals, but not only that, again, back to the data quality-wise, when you apply these principles correctly, right, there's good data showing that as you passage these tumors in animals over successive passages, there's drift in the tumors, and they get further and further away from the original patient material that you collected. So when you use OncoRat, there's no need to passage any further than passage 1 or 2, depending on how you number these. And so it's very low passage number, and you can always go back to your bank, a very well-established bank, and you would always be working with the passage 1 tumor for your studies. So your data quality, again, is going to be better, and hopefully translates to the clinic better. So yeah, this is again, right, expansion just graphically showing, how bigger the tumor grows, how, the passage, elimination of the passages. And again, not only lower passage for quality, lower number of animals used, but for establishment and expansion, the time, again, is highly reduced to establish a PDX bank. The tumors grow bigger. We don't have to go through successive passages. And also the tumors, the growth rate is faster in the rat than in the mouse. So with that, I will wrap up. Thank you so much, and turn it back to you, Megan.
Great. Thank you very much, Dr. Jamling. As I mentioned before, please go ahead and continue to submit your questions at the bottom in the Q&A section. My first question that I've received is for Dr. Jackson. Sam, how can individual researchers ensure they aren't reproducing a previously completed study?
Yeah, this is a really good question, and it's quite hard to do. I mean, I think my first answer would be do a literature search, which is pretty obvious, I guess. But as we all know, a lot of, especially negative studies, aren't reported at all. This is something that we've been talking to various people about as to how we can start to ensure that data from that comes out negative in a study is actually out in the public domain, so that another researcher can see that someone's already done that experiment and not repeat it. It is a difficult one, because unless it's been published, you're unlikely to be able to find a lot of information on it. I think we are usually, as scientists, embedded in communities of people who work on similar things to us. So certainly keeping an eye on what your colleagues are doing, to as much as you can, is a good idea too, and sharing information. But obviously, some of that is going to be proprietary. So yeah, I think that the key way to do that would probably be literature searching.
Fantastic, thank you. The next question is for Dr. Jamling. How do you get the OncoRat for research? Can you buy them outright, or does Hera do the studies for you?
Really good question. Thank you, Megan. So Hera is a CRO. It's a small CRO. So we do services in the OncoRat ourselves here on site. But we also do commercially sell the SRG OncoRat. And it's a timely question, because third quarter this year is when we started making the rats available from Charles River. So we derived a clean colony at Charles River. It's established there. So if you're interested in getting the animals, please get in touch, and the animals would ship from Charles River to your facility. And of course, if you're interested in doing studies with us, we have the animals here ourselves, and we offer a preclinical CRO service in the animals.
Fantastic, thank you. The next question is for Dr. Jackson. How do you collaborate with other 3Rs groups in order to maximize your overall impact?
Yes, so there are in various countries, certainly in Europe and in the US, dedicated 3R centres that work on various different aspects. In actual fact, while we collaborate as widely as we can with those other centres, it tends to be that they specialise in one thing or another. So a lot of the focus is on replacement technologies and developing technologies to try to replace some of the animal studies. Essentially, I feel that the other 2Rs are sometimes a little less kind of dwelled on, if you like, and actually it's sometimes difficult to create a technology or something saleable that you can then attach to reduction or refinement. But we have lots of examples of those in our portfolio where researchers have been able to do that. I think that there is great value in, for instance, the US and Canadian societies which have been set up, getting through to the grassroots of science in those areas. I think that these principles of the 3Rs are pretty well established all over the world, but seem to be more ingrained in scientific thinking in the EU than in North America at the moment. And that's entirely my own opinion, but that's the impression I get. So yeah, collaborating with the groups that have been set up around the world is a really important way to try and get the best practice into labs.
Fantastic, thank you. And the next question is for Dr. Jamling. This person is wondering if you have immune system humanized rats for immune oncology studies.
That's an excellent question. So yes, it's an obvious route to go because of those 10 times volume of the blood in the rat. And so we are working on it. We have some really exciting data on PBMC and BLT humanization in the rat. We're not quite there to offer services in the humanized immune system rat yet, but we are getting close. It's an R&D project right now internally. So we are getting engraftment of PBMCs. We're getting engraftment through the BLT method. Now we need to get it consistently, the consistent engraftment, and also getting the engraftment levels higher. So we are optimizing the procedures to make it more consistent. It's quite variable right now. We are getting engraftment, but it's not quite ready to be offered as a commercial service yet. But if you have interest in humanized immune system rat and are interested in collaborating with us and furthering this R&D project, we would be very interested in hearing from you.
Fantastic. And for those listening in, you can also contact Hera BioLabs through our platform at scientist.com. And then one final question for Dr. Jackson. Can you describe the CRACKIT program in a little bit more detail and explain who can participate?
I'd be very happy to do that. So CRACKIT, I mentioned earlier, is our open innovation scheme. So essentially what we try to do with CRACKIT is to take technologies that may have been developed, for instance, in an academic setting, and give them the push that they need to get to a point of commercialization where they can then either be sold on, or a company can be set up to perform a service or give the product. It's a competition-based scheme, and it's essentially a funding scheme, but it's very closely project managed by ourselves. And so usually there are two types of challenge, but the larger type of challenge is a two-phase challenge, whereby we invite applications to a problem which has been set by industry usually. So to answer a specific unmet need in industry, they will set a challenge with us, and we will put that challenge out to tender. And so the people that apply for those are consortium-based groups, usually, because the challenges are quite large. Those applications will come in. We usually choose 3 or 4. of those applications. Actually, I should say that's done by an external panel, which are then funded for a phase 1, 6-month proof of concept study. And then those people come back in and sit in front of the panel again and show what they've done over 6 months. And usually one group is then funded for 3 years for usually around £1 million for those projects. So these run once a year. We launch them usually in the new year, and consortia can then apply for them, or indeed contact us to ask us questions about the challenges. So those challenges are usually fixed to a fairly thin area, and they will be specifically around developing a technology usually. But as I said, these are consortium-based problems, and so usually these challenges need different expertise being brought together to be able to actually answer them properly, which is one key facet of this. The funding is currently available in the UK and the EU, but certainly US partners can be on those projects as well. And all the details about this are available on the NC3R's website, and actually Crackit has its own dedicated website, which we call our innovation platform. And so all of the projects, past and future and present indeed, are on there for you to be able to look at.
Fantastic. Thank you, Sam. And that was the last question. If you have any follow-up questions, feel free to submit them to us via email. And I'd like to thank Dr. Jackson and Dr. Jamling for joining us today. And to all of you, thank you for listening in. Have a wonderful rest of your day.